Hi students, this is Emandri Kumar Rajesh. In this video, we are going to discuss the remaining two types of enzymes which are widely used in biotechnology. They are alkaline phosphatase enzyme and DNA ligase enzyme. And we are going to discuss the types of enzymes which are used to isolate the DNA from different types of cells. First, we are going to discuss the types of enzymes which are widely used in biotechnology. Let me tell you, alkaline phosphatase enzyme. Alkaline phosphatase enzyme first. So, alkaline phosphatase is an enzyme which is used to remove 5'N phosphate. Alkaline phosphatase is an enzyme which can remove the 5'N phosphate from DNA and RNA. DNA and RNA. Then it will able to remove the phosphate from nucleotide and proteins. That alkaline phosphatase enzyme can remove phosphates from the nucleotide and proteins in the DNA and RNA. So alkaline phosphate can remove 5'N phosphate from DNA and RNA, and it can also remove phosphate from nucleotides and protein. Then that alkaline phosphates are more active at alkaline pH. Alkaline pH. That is why that enzyme is called as alkaline phosphatase. So this enzyme is more active at alkaline pH. That is why they are called as alkaline phosphatase enzyme. Then I am going to tell about the source of alkaline phosphatase enzymes. Source of alkaline phosphatase enzyme. The first source is the bacterial alkaline phosphatase. Bacterial alkaline phosphatase enzyme. That means that alkaline phosphatase enzyme is derived from the bacteria E. coli. E. coli. So the bacterial alkaline phosphatase enzyme is derived from E. coli, and this enzyme is more active. This enzyme has more active. Then the main disadvantage of this enzyme is it is very difficult to destroy after at the end of dephosphorylation. So this enzyme is very active. The disadvantage is this enzyme cannot be destroyed easily at the end of dephosphorylation reaction, and that is the main disadvantage of bacterial alkaline phosphatase. Then the second one is calf-industine alkaline phosphatase. Calf-industine alkaline phosphatase. So this enzyme is purified from bovine intestine. Purified from bovine intestine. So alkaline phosphate is widely used in biotechnology lab because it can be easily destroyed by protease digestion or by heat. So these enzymes can easily digested by protease enzyme or heat treatment, but it has less effective. It has less effective when compared than the bacterial alkaline phosphate, but it is widely used in biotechnology labs due to the reason it can easily digested by the enzyme protease or by heat. Then the third one is shrimp alkaline phosphatase. Shrimp alkaline phosphatase. So this alkaline phosphatase is derived from the shrimp, the cold water shrimp. That is why it is called as shrimp alkaline phosphatase. Now I am going to tell about its uses. Alkaline phosphatase uses. Let us talk about how the alkaline phosphatase enzyme is used in DNA manipulation. For example, we are going to make a recombinant DNA. So in order to make a recombinant DNA, first we have to select a plasmid. So it is a plasmid. Plasmid is nothing but a extra chromosomal material. So this plasmid, in the plasmid we are going to make a cut by endonuclease, to insert the gene of interest in the plasmid. So the endonuclease make a cut in the plasmid. Then after that cut, this plasmid become linear. The plasmid become linear and it contains 5' end with a phosphate group and 3' end with the hydroxyl group. In this situation, again this plasmid can, this DNA segment can, the DNA segment or molecule can self-ligate. So in order to prevent the self-ligation, the alkaline phosphatase enzyme is used to remove the phosphate group because it contains 5' end with phosphate group and it contains hydroxyl group in 3' end. So it can again self-ligated. So in order to prevent that self-ligation, we use the alkaline phosphatase enzyme to remove the phosphate from the 5' end. So now the self-ligation is prevented. Then we can add the gene of interest. So it also contains 5' end and 3' end. Then the gene of interest is ligated by the enzyme DNA ligases. So this gene of interest is ligated with the plasmid with the help of enzyme DNA ligases. Then I am going to tell about the DNA ligase enzyme. Let me explain how the DNA ligase enzyme is used in making recombinant DNA. Here it shows the vector DNA, that is the plasmid DNA, and it is a gene of interest. So the DNA ligase make a ligation between the vector DNA and the gene of interest by creating the covalent bond between vector DNA and gene of interest, and that process is called as ligation. So the DNA ligase creates the covalent bond between the vector DNA and gene of interest, and that process is called as ligation. Then ligation process involves 3 steps. They are self-adenylation of DNA ligase and adenyl group transfer to DNA and phosphodiester bond formation. First, the DNA ligase get self-adenylated. So here the DNA ligase get self-adenylated and it uses for that process 1 ATP. For that adenylation process, it uses 1 ATP molecule. Then that adenyl group transfer to the DNA. So that adenyl group transfer to the DNA and it get attached to the 5' end which contains the phosphate. So the adenyl group transfer to the DNA. Then it creates the phosphodiester bond between these 2 ends. It creates the phosphodiester bond between the 5' end and 3' end. And after the creation of phosphodiester bond, this adenyl group get detached and released as AMP. That means adenosine monophosphate. After the formation of phosphodiester bond between the 5' end and 3' end, this adenyl group released from that phosphodiester bond and released as AMP, that is adenosine monophosphate. This is how the DNA ligase create the ligation between ligation between the gene of interest and vector. Then I am going to tell about the enzymes which are used to isolate the DNA from different types of cells. So please wait a minute. Let me tell about the types of enzymes which are used in isolation of DNA from the cells. That enzymes can isolate the DNA from by digesting the cell wall and the cell. That is why the enzymes are called as lysis enzyme. That is, the enzymes are called as lysis enzyme. Suppose if it is a plant cell, the plant cell contains cell wall and it is made up of cellulose and pectin. So we have to use the enzyme cellulase and pectinase to digest the cell wall. Then it contains plasma membrane and it is made up of proteins. So we have to use protease. And it contains phosphate. So the plasma membrane is made up of protein and phosphate. So we have to use lipase to digest the phosphate. After that digestion, we can get the DNA. After the digestion, we can isolate the DNA from that cell. Suppose if it is an animal cell, the cell wall is absent in animal cell and it contains plasma membrane. So we have to use protease and lipase to digest the plasma membrane. Suppose if it is a fungal cell, the fungi cell wall is made up of chitin. So we have to use that enzyme chitinase to digest the cell wall of fungi. Then it contains plasma membrane. So we have to use the enzyme protease and lipase to isolate the DNA from the fungi. Suppose if it is a bacterial cell, the bacterial cell wall is made up of peptidoglycan. So we can use that enzyme lysozyme. So we can use this type of enzyme to isolate DNA from the cell. So that enzymes are called as lysis enzymes. So that's all about today's video. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.